All right, hello and welcome everyone to the Reclone Network March Symposium, Manufacturing Diagnostics 101. We're so glad to have you all here with us. My name is Kayla, I'm one of the organizers, and I'm sure you'll all have seen the emails from me. But we're really excited to have you here. We've got two really exciting keynote speakers with us today, one discussing RPA and CRISPR technologies, and the other discussing the history and challenges of the first pharmaceutical company recovered by its employees. Before we get started, just a bit of housekeeping. As I'm sure we're all familiar with Zoom by now, I just want to remind you to stay muted if you're not speaking and also be mindful of background noise. Feel free to use the chat to ask any questions throughout the symposium. We'll do our best to monitor the chat to make sure all questions are answered, so please do be patient with us. And also, if there are any technical issues, please be patient. We will work those out. Let's see, lastly, as you will have heard, we are recording these presentations. So if you are asking, or if you do ask a question and you don't want to be in the recording, please send me a private message and we will clip you out. All right, any last things? Last note, I think I've put that in the email as well, but additionally, you can join our Reclone network to stay connected with us and discuss your work at upcoming meetings and within our forum. I'll post the link again into the chat now, so you have it again. And why don't we crack on with our first keynote? So our first keynote is Dr. Aimorn Homchan. She is a postdoc researcher within Dr. Utamapinant's group at Vidyasiri Medi Institute of Science and Technology. Sorry about that. And they are developing in-house RPA and CRISPR-Cas-based detection. So Dr. Homchan has received her bachelor's degree in chemistry and her doctorate in biochemistry from Mahidol University in Thailand. Her PhD research focused on the biochemical and biophysical characterization of DNA binding proteins involved in DNA repair. Recently, Dr. Homchan's work has been focused on developing in-house RPA and its applications to CRISPR diagnostics, particularly for SARS-CoV2. Here, Dr. Homchan will be telling us about their work using Cas13-based detection of SARS-CoV2 RNA with locally manufactured enzymes. So Dr. Homchan, if you want to share your screen, I will hand it over to you.
Okay, can you hear me clearly?
Yep, sounds good.
Okay. Right, thank you for the introduction. As you know, for more than a year, the global pandemic of COVID-19, which is caused by the novel coronavirus SARC-CoV2, has affected all aspects of our lives. So to control the pandemic disease, a rapid and accurate diagnosis of COVID-19 is essential. And for the gold standard RT-qPCR, it's the most commonly used for the detections of pathogen nucleic acid, including the nucleic acids of the SARS-CoV-2. However, it's costly, labor intensive, and it requires special training and equipment. For several countries, particularly in the developing nations, the access to the diagnostic technology is limited due to the low purchasing ability and disruption of supply chain and lack of the infrastructure. So alternatively, the isothermal amplification combined CRISPR-based assay become the method of choice to detect SARS-CoV-2. So this method is highly sensitive and precious and has potential to be applied for massive screening due to minimal instrument requirements, simplicity, and low cost. So yeah, our effort is towards local productions of diagnostic technology for infectious diseases such as COVID-19, as well as other diagnoses, like for detections for critical targets for like food safety or environmental monitoring. So yeah, the one of specific goal right now in our group is to locally produce RPA reagents to lower the cost of deployable Cas13 detection technology and make it more accessible for everyone, especially for in the developing nations. So yes, in the last August, our group reported a clinical validation of a Cas13-based assay for the detections of SARC-CoV2 RNA. So here we rely on the Sherlock method, which has been established by our collaborator Homchan and co-worker at Broad Institute of MIT and Harvard. So here, the figure here show how the Sherlock works. So so basically here there's like the regions of interest that we would like to detect in the SARC-CoV2 RNA. We use the reverse transcription RNA to like reverse transcribe and amplify RNA to the DNA. After that, DNA is converted to RNA by the T7 RNA polymerase. After that, the Cas13 complex with the CRISPR RNA target on the amplified RNA and trigger the characteristic cleavage activity that can cleave the RNA reporter. And after that, the RNA reporter can be captured on the lateral for strict or can be visualized through the first and readout. However, today I'm going to mainly talk about the productions of the RPA. The RPA stands for recombinase polymerase amplification. It is an isothermal amplification method that is remarkable due to its simplicity, sensitivity, and selectivity. For the RPA, it can rapidly amplify either DNA or RNA targets at like ambient and constant temperature ranging from 22 to 35 degrees Celsius, and it can amplify the targets completely within 1 hour, and it's compatible with multiplex. This means if you would like to amplify more than one target, it's okay if you use the RPA. RPA doesn't need a thermal cycle, and so it can be used in the points of care test, particularly in the low resource settings. Yes, and our goal right now, we're trying to produce the in-house RPA to be high sensitive and reliable. And the benefits of the in-house RPA, including not only cost saving, but also flexibility to adjust all the components for improving the performance of the RPA for the particular work. So yeah, here is the mechanisms of the RPA. The T4UVSX recombinase assisted with the UVSY recombinase loader form a complex with the primer. After that, this complex will scan into the double-stranded DNA, searching for the homologous sequence. Once the homologous is found, the primer are inserted by using the standard displacement activities of the recombinase. And the displaced single-stranded DNA is stabilized by a T4 GP32. It is a single stranded binding protein. And after that, recombinase is dissociated from the strand, allowing the elongation of the primer by the BSU DNA polymerase. Next, the newly generated DNA strand is used as the next, as in the template in the next route of the RPA. So that's all the cycles of the RPA mechanism. So our group receives the RPA plasmids from our collaborator Shelley Grubert at University of Cambridge, and Michael Crone and Paul Fremont at Imperial College London. So here are the QR code that can direct you to the benching website in which all the sequences are deposited. So right now we expressed the proteins components of the RPA in the E. coli and purified to be like a more than 95% purity, as you see in the gel. And we follow the standard protocol reported by the Pippenberg and co-worker who has been established the RPA technology for the nucleic acid amplification, except that we use a PEG instead of the Carbovac 20M as a clouding agent. Yes, we tested the activities of our in-house RPA through the amplifications of the S genes of SARC-CoV2. As you can see here, the results show the clear amplification band here across the different concentrations of the DNA input, indicating that our in-house successfully to amplify the DNA of the S gene of the SARC-CoV2. As you can see here, monitoring of the RPA product through the agarose gel was simple. However, sometimes we couldn't differentiate between the false positive due to primer dimer and the two positives of the S amplicons, seems like the two bands were close to each other. So we next decided to monitor the activity of our in-house RPA through the CRISPR-Cas13 activity using the lateral force strip. So here's a scheme show how strip works. So actually in our experiment, there were two reporters. One was biotin's fluorescein functionalized Cas13A reporter, and another one was deoxygenane's fluorescein functionalized RNase reporter. So basically, Cas13A could keep only the biotin's fluorescein reporter, whereas the RNase could keep both reporter. For the negative result, only band at the control position is produced when the anti-fluorescein conjugated gold nanoparticle binds the intact biotin fluorescein reporter and stuck at the septoidine position. And for the perceived for the perceived result, the Cas13A can cleave the biotin fluorescein reporter, allowing the presence of two bands in the strip. Whereas T2 is the bindings of the anti-fluorescein conjugated gold nanoparticle with the deoxygenate fluorescein reporter. And the T1 is due to the bindings of the anti-fluorescein conjugated gold nanoparticle bind to the cleaved fluorescein and stuck at the anti-rabbit IgG. And for the RNase contamination result, the RNase can cleave both reporter, and it's going to present only the band at the T1 position due to the binding of our anti-fluorescein conjugated nanoparticle with the cleaved fluorescein reporter. So here's just the actual result. As you can see here, the commercial RPA was sensitive in the detections of the S genes of the SARC-CoV2 as low as 10 copy per microliter, whereas our in-house RPA could detect when like 1,000 copy per microliters of DNA was present. However, our in-house RPA version was the sensitivity of the in-house RPA was comparable to the commercial RPA when we added the additive inside the reaction. So it was an impressive result. So not only using the DNA as the input, so here we combined the reverse transcripted with our in-house RPA and additive. And as you can see here, the sensitivities of the in-house versions was comparable to the commercial RPA in the detections of the end gene of the SARC-CoV2. So yes, I'm very impressed, really impressive of our in-house RPA right now. So yeah, besides the additives, I would like to tell you there are more parameters of the RPA that should be accessed for the optimization, such as like enzyme concentrations, primers, temperatures for the incubation, and the amount of magnesium, pH, or screen mode additives. Like you can praise allowed all the components in the in-house RPA for improving your performance of the RPA for particular works. So, ongoing work right now. So we are optimizing the in-house RPA by like varying the enzyme concentration, and also we like screen more the additives, and we try to lyophilize our in-house RPA for solid at ambient temperature and for easily set up and transportation. And why we are optimizing the in-house RPA, we also optimizing the multiplex detections of the Cas13 based detections of the two genes in the SARC-CoV2 as an end gene, and assessing this assay on the clinical samples. So yeah, here is the screen show how multiplex RTRP, multiplex Cas13 based detections of the two genes works. So basically, we decide two sets of primers that's specific to the different targets of interest, and we use the transcription RPA to reverse transcribe and amplify RNA to DNA, and then we use the T7 RNA polymerase to like convert DNA to RNA, and here we use the collateral Cas13A and Cas13B, which can cleave the target independently. And here we demonstrated that Cas13A and Cas13B function orthogonally in the multiplex detection. As you can see here in the left-hand side here, the activities was due to the Cas13A. It could detect only the S gene, even though the end is present. Whereas the right-hand side, is the activity due to the Cas13b. As you can see, it could detect only N genes, although the S was mixing in the reactions. Moreover, we were trying to create the simple device to detect the multiplex reactions by eye or smartphone. So here, basically, we selected 244 that's basically these 244 are mutually excited by the single blue light source, and then we use the plastic filter to allow only emission spectrum for those 244 can transmit, and then we can easily read the result by eye, or we can use our smartphone to capture the results of the multiplex reaction for quantitative analysis. So yes, that is for my talk. So here's main people who are working on the local RPA production and specific Cas13 with detection. Dr. Shesit Uttamapinan, who's our group's leader, Dr. Danaya Pakotipapa from Faculty of Science, Melbourne University, and our CRISPR team, Kanokpon Turadan Zuraset. And we thank our strong collaborators from Faculty of Medicine, Sirat Hospital, Mahidol University, Dr. Nevin Hong Thongkam for contributions of the RNA sample. And we thank all the CU lab members for encourage us to produce this impressive work. And we thank all these organizations for funding and financial support. So yeah, that's it from my talks today. Thank you.
Thank you, Dr. Homchan. That was a great and very interesting presentation. If we have any questions, feel free to ask.
What's the temperature you used? I missed because you mentioned anything from 20 to 45 degrees is okay, but what is the optimal temperature?
Yes, I think this depends on your amplicons. So basically, sometimes we found that the temperature like 39 degrees Celsius is much better than like 37 degrees Celsius for the particular amplicon that you would like to detect or you would like to amplify. I think vary the temperature starting from like the ambient temperature 37 degrees Celsius to like 42 degrees Celsius would be better. Mm-hmm.
Thank you.
Yep.
Have you tried making your own enzymes?
Excuse me, making my enzymes. You mean Cas enzymes or RPA?
Yeah, well, for your assays, yeah, the RPA assay, producing your own enzymes.
Yes.
Yes, okay.
Yeah. We express and purify by own and trust us compared to the commercial one.
Wonderful.
Thank you.
We have time for one more question.
Do you have any test of the quality of those enzymes, like specific activity?
Interesting. Yeah, we haven't tested their separately activities. It would be great to test the activity separately. Yes, thank you. And we just like a mix inside like a cocktail and tested the activity. Thank you.
Lovely. Thank you so much, Dr. Homchan. You can stop sharing your screen now. For further discussion on RPA, please do join the breakout room #1, best practices for RPA, and we can continue that discussion later in the symposium. Next up, we have our lightning presentations. So Lisa, please take it away.
Thank you, Kyla. Hello, everyone. Thank you all for joining. So we will have 3 short lightning presentations now. So we are on a very tight schedule, so we don't have time to do questions straight after the presentations, but you can ask them on the Reclone forum or later in the discussion breakout rooms. So yeah, our first presentation will be given by Brooke from the Ethiopian Biotechnology Institute, and he will speak on the development of LAMP instrumentation using beeswax, using engineered beeswax as a thermal buffer. So, Brooke, if you want to share your screen.
Yeah. Here do you hear me? Yeah.
Yes, I can hear you.
Yeah. See, where is it? Share screen. Oh, Chrome tab. Okay. Is it visible now?
Yes, it is visible.
Yeah, thank you for the opportunity. My topic is developing robust isothermal heat bath for LAMP using beeswax nanocomposite as a thermal buffer. WHO recommends for countries like my country, like in the developing country, this so-called assured criteria: affordable, sensitive, and specific, user-friendly, rapid, and equipment-free, and deliverables. To end user, in this regard, LAMP is a preferred technique because it's simplified in instrumentation, and it can amplify to 1 billion copies in 30 minutes. Primer design and multiplexing is also possible, and it can be made visual detection in user-friendly, and time to result is came as short as 30 to 45 minutes, and it doesn't need such a well-equipped lab facility, and it can go to point of care facility. And for that, for developing our LAMP isothermal heat bath, we consider the assay parameters, and on the other hand, the operating environmental condition. The assay parameters is obvious. Optimal assay temperature has to fit with optimized time. Assay tolerance fluctuation, temperature fluctuation is serious consequence on the sensitivity, and it has to be in a minimum fluctuation range, minimum holdover time for the amplification, and quick warm-up time. In countries like Ethiopia, with altitude varies from -140 below sea level to 4,900 above sea level, wherein the ambient temperature varies between average between 4 degrees centigrade and 43 degrees centigrade, and we need to use this instrument, so it has to be robust in all this temperature range. For that, our heat block component power source adjustable, we made it an adjustable output, and with active feedback control, and for phase change as a temperature regulator, we used beeswax because of its higher heat capacity at melting point, which coincides with amplification temperature, which is 60 to 63 degrees centigrade, and effective thermal insulation. and some of the thermodynamic requirements are many for this phase change material, but the highlighted one, melting point temperature must coincide, high latent heat of fusion, and high thermal conductivity. In the chemical properties, reversible freeze and melting cycle without degrading its chemical property after several loading and unloading cycles. And for that we selected beeswax. It has excellent melting point. As you can see, at 63 degrees centigrade it has a deep, and this equivalent to 230 kilojoule per kg. When you compare it with to do these things with water baths, it is 55 times robust. That's why we use that one. And the problem with the beeswax, it has low thermal conductivity, so we enhance it by making use of copper oxide nanoparticle, which we prepare through sol gel preparation, and we confirmed that with XRD diffraction, which coincides with the major peaks. And in addition to that, we inserted through fractal geometry, as you can see here, the fin, 9 fins, which protrudes directly into the beeswax, and this is how we assembled it in right manufacturing workshop. And with this one, we developed and we tested, even in the absence of heat supply, with the lid opened, the buzz exposed to the ambient temperature we could maintain for 18 minutes without power supply at that isothermal temperature. But with the presence of power, it could continue to more than 2 hours. So this leads us to the conclusion that the buffer is very convenient for using as a lamp thermal buffer, and some optimization are added. If we add some optimization, we can use it as a microfluidic or lab-on-chip format, using to use this engineered beeswax. Thank you. This is, if you do have any question, I'm ready to elaborate. Of course, we tested it for SARS-CoV2 test using a commercial kit from New England Biolab, warm start lamp M1800 visual detection. It could correctly identified the positive from the negative. Thank you.
Thank you very much. That was very interesting. Unfortunately, we don't have time to take questions right now, but you can ask them later in the discussion groups. So next we will have China from Munster Technology University presenting on low-cost open source hardware